Hi. So, in previous session we have looked at about the TLR, and we have seen various kind of PAMs, or I will say the array of PAMs are sensed by this TLR, which is expressing on variety of innate immune cells such as macrophages, dendritic cells, basophil, and mast cells and B cells as well. Okay. So, upon sensing they induce inflammatory cytokine, that you are very well aware. They are also inducing type 1 and type 3 interferons. Okay. Now, let us look at what kind of signaling is operated. Okay. And in this session we will discuss in great length about the TLR signaling. Okay. So, this slide you are well remember, as I have shown you in previous session, that these TLRs are basically localized over cell surface, and these TLRs are also expressed in endosomes. Okay. And here if you see very carefully, there are only few adapters molecules are there, which basically play an important role in signaling. Okay. So, if you see the TLR4 basically uses 4 adapter molecule. Okay. The first adapter molecule, if you see, this uses TRAP, which is also known as MAL. Okay. In previous session I have discussed this MyD88 and MAL, which is also known as TRAP. They have a TIR domain. Okay. So, TLR4 uses this MyD88 as well as TRAP or MAL. In addition, TLR also uses another very important adapter known as TRAM and the TRIF. Okay. So, TLR4 uses all adapters of the TLR signaling. On another hand, if you see TLR2 along with 6, okay, and 2 along with TLR1, which makes a heterodimer. So, this uses TRAP or MAL along with MyD88 in order to activate the signaling. And rest of other TLRs basically uses MyD88 except TLR3. If you see, TLR3 uses only one adapter, that is TRIF. It is quite complicated for you, right? But do not worry, it is not too complicated. If you will see subsequent slide, then you will understand very quickly and easily. Okay. Anyway, so upon recruitment of these adapter, basically there will be a downstream signaling, and this downstream signaling result to the production of pro-inflammatory cytokine and type 1 interferon. Now, here just I want to take you again upstream. So, how this sensing is taking place? Okay. So, sensing is as I told you in previous session, this leucine rich repeat is playing a very important role in sensing. Basically, this makes a horseshoe like a structure. Okay. And this horseshoe like a structure is basically playing important role. So, what is happening when there is a PAMP or if there is a or any ligand PAMP or any ligand, when it comes in contact with the cell, then what happens? Basically, there is a oligomerization or clustering of these receptors are taking place, and this clustering will basically trigger the recruitment of these cytoplasmic adapter molecules. And once this cytoplasmic adapter molecules are recruited, then the cascade of signaling is taking place and finally result to the transcription of pro-inflammatory cytokine and type 1 interferons. So, since the signaling is little complicated, we study this in much more simpler way. We study the TLR signaling in two major part. One, we call it as a MyD88 dependent signaling, okay, and another we call it as a TRIF-dependent signaling. MyD88 is basically myeloid differentiation primary response gene 88. This is a full form. Anyway, you need not to worry about the full form, just remember the MyD88. And basically TRIFs stand for TIR containing adapter inducing interferon beta. So here you can guess that this adapter is playing important role in induction of interferon betas. Okay. And MyD88 dependent signaling is basically operated in these cell types. As you can see, there is a macrophages, dendritic cells and plasmacytoid dendritic cells. TRIF dependent signaling is mainly operated in macrophages and dendritic cells. Okay. Now let us take up this MyD88 dependent signaling. So here you can see that TLR1 and 2 and 2 and 6, they make a heterodimer, and this heterodimer basically activate the signaling. Whatever TLRs which you are seeing in this image in a monomeric form, ideally it is not in monomeric form. In cellular condition it is either dimer or oligomer. I have told you there will be a clustering of receptor. Okay. So once these receptors, once this ligand bind to the leucine rich repeat, then that will cause the conformational change in TIR domain. Okay. And this conformational change basically recruit the adapter molecule MyD88, but in case of TLR 1, 2, 2, 6 and 4, there will be additional adapters will be there, that is MAL or TREP, and upon binding with or recruitment of MyD88, there will be a recruitment of various other signaling molecules which consist of a family of IREC family protein, which is mainly IREC 1, 4, and there is some more IREC are there. One of the IREC is IREC M, which is basically a kind of negative regulator for this MyD88 dependent signaling. Anyway, so let us move on. So IREC will be recruited, and then there will be a recruitment of this TRAF6, and this complex or signalosome basically activate, there will be many more proteins are there. For simplicity, I am showing you in a very simple way. The overall aim of this signalosome is to activate the IKK complex. Okay, and once this IKK complex get activated, then it will phosphorylate, that will activate the kinase activity. Okay. So this kinase basically activate I kappa B, and once I kappa B get phosphorylated, then what is happening, then there will be a ubiquitination of this I kappa B, and this ubiquitinated I kappa B is degraded by the proteasome. Okay, and NF kappa B will be free, and this NF kappa B will be translocated into the nucleus, and this active transcription factor transcribes the gene for pro-inflammatory cytokines, okay, or inflammatory cytokines. So this is the one way by which the signaling is taking place. Another way is this signalosome, this complex of protein can also activate the MAP kinases. Okay, as you know, you probably studied this MAP kinase pathways. There are various molecules like JNK, P38, ARC. All these guys will be activated, and that will also induce the synthesis of pro-inflammatory cytokine. But in PDCs, which is which we call it as a plasmacytoid dendritic cells, in those cells there is a little different pathway, not little, quite different pathway is operated, and this different pathway basically plays a very important role in production of robust amount of type 1 or type 3 interferons. Okay. So, what is happening in PDCs, the MyD88, which is an adapter molecule, this directly interact with transcription factor and phosphorylate it. So, here try to understand, the whole signaling pathway is kind of cut off, and this MyD88 basically directly phosphorylate the transcription factor known as IRF7. Okay. And this IRF7, the phosphorylated IRF7, basically once they get phosphorylated, they make a dimer. Okay. Phosphorylated IRF7 make a dimer, and then it is translocated into the nucleus, where there will be a transcription of type 1 and type 3 interferons. Okay. So, in that way, this whole MyD88 dependent pathway is operated. Now, let us look at another very important component of the pathways, that is TRIF dependent pathway. So, TRIF is an adapter molecule which is used by two major TLRs. One is TLR4 and another is TLR3. TLR3 exclusively use TRIF in order to induce the pro-inflammatory cytokine as well as the type 1 interferon synthesis. Okay. Here you can see. So, upon sensing of this double stranded RNA molecule or poly-IC, okay, which you have seen in previous session, there is a poly-IC. Poly-IC is it's basically a polymer of inosine and cytidine. Okay, and this polymer of inosine and cytidine is we use it as a double stranded RNA. Okay, and if you introduce this double stranded RNA, this poly-IC inside the cell, then it will mimic like a virus infection. Okay, and if you stimulate the appropriate cell like macrophages with poly-IC, then it will induce or activate the TLR3 pathway. Okay. So, here you can see that TRIF is used by TLR3 and TLR4. Of course, in case of TLR4, this also recruits another molecule known as TRAM. Okay, and TRAM and TRIF is used by the TLR4. And this TRIF can also activate this IREC and TRAF6 in order to induce the synthesis of pro-inflammatory cytokine via activation of NF-kappa B, as I have explained you in previous slide. TRIF can also activate the MAP kinases in order to synthesize the pro-inflammatory cytokine, and TRIF basically activate a kinase which we call it as a TBK1 or IKKI. IKKI is also known as IKK epsilon. Okay. So, this kinases, once this kinase get activated, so these kinases phosphorylate IRF3, and this phosphorylated IRF3 again becomes a dimer, and then it is translocated into the nucleus in order to synthesize the type 1 interferons. Okay. So, this is a very simplified view of this whole TLR signaling. Now, I will just summarize this TLR signaling with one very simple slide here, in order to remember that what are the adapters used by the various TLR. If you remember that TLR2 along with 1 and 6, they use MyD88 or TREP or MAL, and TLR4 uses all adapters. Okay, that is MyD88, TREP or MAL. This is the name of same molecule, TREP and MAL. Okay, and it also uses TRIF and TRAM, and rest of TLR uses only MyD88. TLR3 uses TRIF. Okay. So, this is the kind of summary of TLR and its adapter. Now, let us look at what are the inflammatory mediators and type 1 interferon, although I have shown this slide, but I will quickly go through. So, this also induces vasoactive amine. Vasoactive amine, I am not very clear about that how this this is involved in production of histamine and serotonin, but I am sure they are upon activation of TLR there will be a activation of NF kappa B, and that NF kappa B must be triggering some enzyme which is needed for the synthesis of histamine or serotonin. But I am not very well aware and I cannot say how it is operated, but I am very well aware about that synthesis of escanoid. So, there is an enzyme, if you remember, there is a cyclooxygenase, okay, which we call it as a COX-1, and there is a another cyclooxygenase which we call it as a COX-2. So, this COX-1 and COX-2 is induced upon the stimulation of TLR, okay, and in that way this inflammatory non-proteinaceous inflammatory mediators are synthesized. Bradykinin, most likely it is also derived by or derived through NF kappa B mediated signaling. And there are variety of cytokines like IL-1 beta, IL-6 and TNF. So, they are also playing they are synthesized upon activation of NF kappa B. So, these are the inflammatory mediators. In addition, there is a type 1 and type 3 interferon. Type 1 interferon, which is basically consists of interferon alpha and has a 13 member and single interferon beta. So, they are basically synthesized in presence of transcription factor IRF7 and IRF3. Okay. There is a set of interferon which we call it as a interferon inducible gene. So, this is basically IP10, rentis, and there are so many. Okay. So, this is also playing a very important role in antiviral immunity. Type 3 interferon, which is consists of 3 members and it is also known as interferon lambda. It is also induced through these transcription factors. So, there is a family of transcription factor known as interferon regulatory factor, okay, and there are so many members. Mainly these two members IRF3 and IRF7, they are playing important role in synthesis of these interferon, and some are playing important role in production of inflammatory cytokines. Okay. So, what type 1 interferon is doing? It's interesting. So, basically it induces hundreds of genes, and all these genes are playing important role in antiviral immunity. Here you can see there is a oligoadenylate synthase. Basically it induces the synthesis of RNase L, and this RNase L basically cleaves the viral nucleic acid, okay, and in that way viral replication can be checked. This can also induce P1 kinase. I am just highlighting few of these molecules, but it induces hundreds of molecules, and this P1 kinase is basically induced in order to shut down the cellular metabolic activity, mainly the protein synthesis, that is through EIF2, elongation factor 2. Okay. It also induces MEX protein in response to the influenza in infection of influenza virus, and it provides some kind of resistance. In addition, this activates a very important group of cells known as natural killer cells, which plays a very important role against virus as well as against transformed cells. Okay. Type 1 interferon activates adaptive immunity, and this can basically increase the antibody production by activating B cells, and it also activates CD8 T cell responses. Okay. It induces the expression of MHC class 1 molecule, and it also play important role in expression of MHC class 2 molecule also. Here I am just trying to give you a more interactive presentation about the role of, or how this interferon and type 1 interferon and inflammatory cytokines work. So, basically if you see carefully, this induces apoptosis of virally infected cells, make healthy cells which is not yet infected with virus. This interferon make those cells resistant to the virus infection, it depresses the metabolic pathway. It reduces the as you have seen, there is a elongation factor 2. If you treat the cells with interferon, then it will depresses the metabolic activity in the cell. Okay. It also prepare and activate the professional immune cells for the for development of virus specific immunity. Here you can see in this cartoon. And inflammatory cytokines are basically a kind of alert system. They activate the whole immunity and make a alert, a kind of alarm in the host. So in that way, these cytokines and type 1 interferons are playing important role in innate immunity. So with this, I will stop here, and in next session I will talk about TLR and disease, and is there is a way to use this TLR knowledge to use it for the therapeutic. So I will cover up these two topics. Thank you very much.